increases faster than the surface area does. And it's really important that you know that if you were given surface area to volume ratios, the biggest ratio is the best. On the test on cells, you had to calculate the surface area to volume ratios. You will not have to do that for this. But if you were given options, like let's say you were given choice A, a cell has a 3:1 ratio. Choice B, it has a 1.5:1 ratio. And choice C, it has a 0.75:1 ratio. And they asked you which of these three cells would be the most efficient. Your answer is going to be the one with the 3:1 ratio. Whichever one has the biggest ratio, the biggest surface area compared to the volume, is going to be your answer. More surface area is better. So make sure you know that. The volume increases faster, that's why cells can't get really big. And if you were asked to pick the cell with the best surface area to volume ratio, you're going to want to pick the one that has the biggest number. All right, prokaryote versus eukaryote. You need to know the main difference between them. First of all, prokaryotes are your bacteria, like archaea and bacteria. Eukaryotes are all the others, all the other kingdoms, and all the other domains, besides the ones that include bacteria. The biggest difference between them is that prokaryotes have no nucleus or no nuclear membrane. In other words, their DNA is not protected. So it would look something like this. Here's your prokaryotic organism, and basically here would be your DNA without a nucleus protecting it. It is in an area called the nucleoid, but there's no membrane around it. Eukaryotic organisms have a nucleus or a nuclear membrane protecting their DNA, and they have lots of organelles, the mitochondria, the rough ER, all that stuff. Why are the organelles important? The most beneficial thing about having all the organelles is that it creates compartments. Meaning, eukaryotic cells can have all kinds of separate jobs going on in each area. When I taught this originally, I gave the example of your kitchen. If you didn't have a way to keep your food cold because there was no way of closing off your refrigerator and closing off your oven and having a sink that water wouldn't go everywhere, you couldn't be as you wouldn't be able to do a lot of jobs. So by having eukaryotic cells having compartments, they can have each of these areas with a different environment, which can allow it to have a better metabolism and get a lot more accomplished. They can be bigger, they can do more jobs. So the mitochondria has a completely different environment than the rough ER, or the smooth ER, or the Golgi apparatus. Every part of the cell forms a different compartment where a different job can happen. And having these compartments allows the cells to be more complex. So that's the reason why all the organelles are so important to making eukaryotic cells more complex than prokaryotic cells. If you were to see a picture, you should be able to identify whether it's prokaryotic or eukaryotic. The biggest thing you're going to look for the nucleus. The nucleus and all these organelles is going to tell you it's a eukaryotic cell. Cell parts. The ones I listed up here are the ones you need to know. So any part that's not here, you will not be responsible for. So you don't see there like gap junctions, you don't see cytoskeleton, you don't see peroxisomes. Anything that's not here you don't need to know. But any of the parts that are here are fair game. You could either be asked to identify them from a picture, or you could be asked for their job. So nucleus, we'll go through them really quick. The nucleus contains the DNA. Keep in mind that the DNA a lot of times is called chromatin. It's not called, they don't necessarily say DNA. If they say chromatin, they're talking about DNA, because that's what the DNA looks like when a cell isn't dividing. It's just like this stringy stuff inside the cell. It's in the plant and the animal cell there. And that's called chromatin. Remember that that's the instructions for making the proteins. So it's a set of directions for protein synthesis. Even though proteins aren't made by the nucleus, the directions for making them are there. The nucleolus contains rRNA, Actually, it makes our RNA. That's what the ribosomes are made of. The ribosomes, I'm not going to identify them. I think everybody knows they're like little dots. And they are the ones that actually make the proteins. The rough ER modifies proteins. The smooth ER has a completely different job. It deals with lipids and carbs. And it also is responsible for detoxification, like in your liver, for example. So the rough ER modifies proteins. The smooth ER makes lipids and carbohydrates, and also is involved in detoxification of drugs. From a picture, the rough ER and the smooth ER look very similar. Here's the rough ER. It has the dots around it. And the smooth ER would not have the dots around it. Lysosome. Lysosomes digest things. Technically, you would not be able to identify a lysosome from one of these pictures. However, you could be asked to identify a lysosome if I showed you what it was doing. So like, let me erase this little area and show you what I mean. So for example, if I showed you a cell eating, like if I told you this is a white blood cell, and I showed it eating a bacteria, phagocytosis, and that bacteria went in and it got fused with something, and then it got digested into a bunch of little dots, and then those little dots got basically exported out of the cell, that would be a good example showing you that the organelle that did the digesting would be a lysosome. But I'd have to like explain what was going on in the picture. You would not be able to identify a lysosome from just a regular picture. But if I explained, oh, this is a white blood cell and it's eating a bacteria, you know, what organelle is breaking the stuff down, a lysosome would be the organelle that does that. So it digests things. And a good example would be, the classic example, are white blood cells digesting bacteria they eat. Vacuole, you don't have to know anything about it in animals, but you should know that in plants, it's right here, that this is what allows plant cells to have so much more volume inside of them. This was a previous test question. Plant cells have a lot more volume inside because they have this vacuole that's storing water. So they have all this water in the vacuole, and it creates pressure, and that's what makes the plant stiff. Golgi bodies, or Golgi apparatus, they are the ones that are going to pack and ship out stuff. For example, if a cell was not just making proteins, but let's say it was making glycoproteins, which is a protein with a sugar on it, the Golgi bodies would be the ones that stuck the sugar to the protein, packed it up, and then shipped it out. And this is what they would look like here and here. So Golgi bodies would pack up things for export. A good example of something, a majorly very good example, would be glycoproteins. A protein that's got a sugar attached to it. That would happen in the Golgi bodies. They would make it, pack it, and then ship it out of the cell. Mitochondria, you should know they make ATP. They are in plants and animals. Don't assume mitochondria are only in here's the one in a animal, here's one in a plant. And then chloroplasts also make ATP and use that. They basically take the sunlight, they make ATP from the sun, and then they turn that into sugar. But chloroplasts can only do that in the light, because that's what photosynthesis is, which is technically the chapter we just did. And they look like this. This is a chloroplast up here with the little lines. So mitochondria and chloroplasts are both involved with ATP, but chloroplasts would only be able to do it in the light. They would actually capture the sunlight, make ATP, and then use the energy to make sugar. Mitochondria break sugar down to make ATP. Mitochondria are working in all cells all the time.